So we should be putting our phones down. We should be stopping talking, please. Because we just end class right now, and you guys could just leave. And there have been times when, in the past, I've just walked out of class. Okay, just say, all right, we're not doing anything today. Just you guys can figure it out yourself. Okay, and I absolutely have done that in college too. So if you don't want to settle down, and you're not old enough to sort of have a break and come in and settle down, then that's on you. In terms of, we are in Unit 4, and I want to just illustrate for you what we talked about today and what we're going to be talking about next week. There are 2 chapters that encompass this first unit, this first exam. The first chapter is chapter 11, and that's going to be on the nervous tissue, and that's what we just started talking about. Then the second chapter is on the spinal cord and spinal tissue and spinal nerves. So in terms of this unit 4 terminology or the vocabulary, I'm sorry, that's not right. Oh, here it is, nervous system vocabulary. I skipped over it. So the question is, have I talked about some of these terms? Have I talked about all of them? No, I haven't talked about all of them. We will get into it. So if you just look at the first, say 10 terms right here, central nervous system, peripheral nervous system, afferent, efferent. So the sensory and motor, integrating presynaptic, postsynaptic, neurons, axons, neurons, and dendrites, right? That brings us all around about chapter 12, term 12. Neurons, neuroglia, soma, nissal bodies, neurofilaments, and cytoskeleton dendrites, right? So all of these things right here we're bringing down when we're talking about axolemma and axonal transport, node of Ranvier. We're all down to close to term 30. Synapse, we talked about receptors and effectors. I didn't really talk about that, and I didn't talk about the next few terms. There's somatic sensory and visceral sensory. We'll talk about that in the lecture. Different types of receptors we didn't talk about, but we will. Anaxonic neurons and multipolar neurons, I talked about those two. And with others, this negative 70 millivolts is the resting membrane potential. The different types of neuroglia I didn't talk about. I will tell you later on what I talked about, but I didn't introduce all 60 of them, or all 6 of them rather. Different channels here. When you see the term channel, I immediately want you to think of an integral protein, and that's all we're talking about. There's gated ion channels, there's ligand gated channels, there's it's called electrical stimulus, triggered channels. There's different types of channels, but at the end of the day, they are simply integral proteins responding to a stimulus. That stimulus then is coming from ACH or other neurotransmitters, so we're just worried about ACH right now. And what is ACHE? That's acetylcholine esterase, so that comes from Unit 3. And when you see the words depolarization, repolarization, that's referring to the movement of sodium potassium in relation to the axolemma, and that's moving either into or out of the cell, depending on which ion you're talking about. And then the sodium potassium pump is the term, so it's term 55, and it's a term we use repeatedly throughout the semester. And this is a term that, understanding this, you'll understand, you need to know it for next year too. That sodium potassium pump is the term, or is the mechanism by which our neurons set up the resting membrane potential. We talked about endocytosis and exocytosis. These last two, IPSP and EPSP. The PSP is postsynaptic potential. The I is inhibitory postsynaptic potential. So what is postsynaptic meaning? After the synapse, the area of the neuron after the synapse. That's postsynaptic potential, and you can either have one that's inhibitory or one that's excitatory, and that happens in the brain. So we'll talk about that later on. So I've used close to 50 some odd terms without even extending this diagram. And so I think this is the last time that I'll show you this type of study habit or study skill. Trying to diagram things and using a single diagram that would have multiple different many different terms that you can put on it is a much more efficient way of studying than doing things like Quizlets or index cards, because it's just much more powerful. And I've said that multiple times throughout the semester. So hopefully you've seen it work. In terms of the nervous tissue diagrams here, I will put them right in the top here, and you can see all diagrams here that I've uploaded in the past. I think this is from a diagram that I did during COVID, and I just kept it up here, right? So on the left-hand side, so this is a picture of, there's different pages that I did, and took pictures of it and did lectures. The same terms that we're using in this diagram are what I just did. There might be some differences, but not anything major. There's also, when you're getting into the structure of neurons and the action potentials themselves, they get really complicated really quickly. It goes from very simple stuff to real complexity in a matter in just a little bit. So I want to just try to give you guys multiple different ways of understanding this stuff. So here's one example. There's 2-minute neuroscience videos.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the divisions of the nervous system. There are 2 major divisions of the nervous system. The first is the central nervous system, which is made up of the brain and the spinal cord. The second is the peripheral nervous system, which consists of nerves that run throughout the body. The peripheral nervous system itself is made up of 2 subdivisions. The first is the somatic nervous system, which contains nerves that carry sensory signals from the body to the central nervous system, and nerves that carry motor signals from the central nervous system to the skeletal muscles. The somatic nervous system is associated with voluntary movement. When you clicked on this video to play it, the signal to depress your finger was sent from your brain to your finger via the somatic nervous system. The second division of the peripheral nervous system is the autonomic nervous system. The autonomic nervous system is sometimes called the involuntary nervous system, and it is involved in regulating the internal environment of the body. It carries signals from internal organs to the central nervous system, and from the central nervous system to the internal organs. In this way, it is involved in regulating things like digestion and heartbeat, which are generally outside the realm of conscious control. The autonomic nervous system can be further subdivided into sympathetic and parasympathetic nerve fibers. The sympathetic nervous system plays a large role in stimulating and mobilizing energy resources, while the parasympathetic nervous system acts to conserve energy. For example, if you're in a frightening situation, the sympathetic nervous system will cause your heart rate to increase, your blood pressure to increase, and your sweat glands to be stimulated. If you're eating a meal, however, and are not frightened, your parasympathetic nervous system will stimulate digestion, increase salivation, and slow your heart rate. Due to these functions, the sympathetic nervous system is often described as being involved in fight or flight responses, while the parasympathetic nervous system is described as being involved in rest and digest responses.
So some of this we didn't talk about today, but most of it we did, particularly the somatic nervous system and then the details of the autonomic nervous system. The details of the autonomic nervous system will be in Unit 5. And so again, there's much more overlap between Unit 4 and Unit 5. I'm sort of making an artificial difference between them. Let me see here. I made an artificial division between them. I just want to watch 2 more of these, and then
Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the neuron. This is a brain. Estimates vary, but right now the best guess seems to be that our brains contain around 85 billion neurons. The neuron is a nerve cell, and it's the primary functional unit of the nervous system. This is a generic image of a neuron. Neurons actually come in all shapes and sizes, but this is the prototypical version of a neuron that you'll often see in a textbook. The structures extending from the left side of the neuron that look a little bit like tree branches are called dendrites. Dendrites are the area where neurons receive most of their information. There are receptors on dendrites that are designed to pick up signals from other neurons that come in the form of chemicals called neurotransmitters. Those signals picked up by dendrites cause electrical changes in a neuron that are interpreted in an area called the soma, or the cell body. The soma contains the nucleus, which contains the DNA, or genetic material of the cell. The soma takes all the information from the dendrites and puts it together in an area called the axon hillock. If the signal coming from the dendrites is strong enough, then a signal is sent to the next part of the neuron, which is called the axon. At this point, the signal is called an action potential. The action potential travels down the axon, which is covered with myelin, an insulatory material that helps to prevent the signal from degrading. The last step for the action potential is the axon terminals, also known as synaptic buttons. When the signal reaches the axon terminals, it can cause the release of neurotransmitter. These purple structures represent the dendrites of another neuron. When a neurotransmitter is released from axon terminals, it interacts with the receptors on the dendrites of the next neuron, and then the process repeats with the next neuron.
So there should be a lot of familiarity with this. And you're more than welcome to look at any of these. Let me just see. I'll just do this more general one. Now this is about neuroimaging and about how you go about looking at the
Neuroscience,
brain.
where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss neuroimaging. I will cover a few of the most common types of neuroimaging: CAT scans, MRI, PET scans, and fMRI. Some neuroimaging techniques allow us to see the structure of the brain, while others allow us to look at brain activity or function. Computerized tomography, also known as computerized axial tomography, is a type of structural neuroimaging. It is usually called a CAT or CT scan. It involves taking a series of X-ray images from various locations around the head. These images can then be combined to construct an image of the brain. The resolution of CT images is not that high, but they can visualize any major structural problems of the brain, like a tumor. Magnetic resonance imaging, or MRI, involves applying a combination of magnetic fields and radiofrequency energy waves to the brain. Hydrogen atoms respond to the magnetic fields and radiofrequency pulses by emitting energy. The MRI machine receives this energy and can tell what part of the brain it came from. A computer can use that information to reconstruct an image of the brain that has high spatial resolution. Positron emission tomography, or PET scanning, is a way of imaging brain function. To do a PET scan, a patient is injected with a radioactive substance that emits positrons, which then emit gamma rays when they collide with electrons in brain tissue. These gamma rays are detected by the PET scanner. Because the radioactive substance was injected into the bloodstream, what the PET scanner is detecting is the movement of blood throughout the brain. Blood flow to an area of the brain increases when that area is active, so PET scanning creates an image that highlights the areas of the brain that are being used most while the person's in the scanner. Functional MRI, or fMRI, uses a similar approach to MRI, but focuses on the different responses oxygenated.
So fMRI is, I want you to listen to this part right here. Oxygenated versus deoxygenated blood. And where is oxygen being used? It's used in the brain. Where in the brain, where in the neurons are they used in the mitochondria? Where are the mitochondria? They're both in the soma and then in the telodendria or the axon terminal. And then if you see areas where they're, in this picture right here, there's red areas and then there's the white areas. Those are different levels of oxygen utilization depending on what you're doing with your brain, what you're thinking about.
Blood make to magnetic fields and radiofrequency energy. fMRI uses what is called blood oxygen level dependent contrast, or BOLD, to identify changes in blood flow in the brain, and thus to identify areas of the brain that are most active. fMRI allows one to image brain function without having to inject anything, and provides high-resolution MRI images at the same time as it provides a functional image.
So the fMRI in particular is most relevant to this unit right here. Why is it? Because if there's blood flow to a neuron and there's lots of oxygen flow to the neuron and the oxygen is being sent by the neuroglia in the brain, then those neurons will be producing more ATP and will be sending action potentials more rapidly. Yes?
What if the whole brain, shouldn't the whole brain be lit up because the whole brain includes all types of neurons, like
So this is differences, when you're talking about fMRI, you're talking about differences or increases through the basal rate. So the fact that it, in other words, so right now your heart is racing or beating, right? Let's just say it's at 60 beats per minute. If you go and run down the stairs, your heart is going to increase in rate. If you say that 60 beats per minute is your resting beating rate, and then you just say that's 0, and now you're up to 70. So now you've just increased it by 10. Right? So that's what they're doing here. They're saying that the brain is getting blood, it's getting oxygen, and it's fine. But what area of the brain is most active? And it's looking at those areas of the brain that are most active. So that's not to say that this area is dead and this area is really active and this is alive. It's saying that this is more active than this area. So that's what you're looking at, okay? So again, it's assuming that the brain is normal and healthy. If there's pathological issues with the brain, then that's a different issue, like a stroke or something like that, or a tumor. Does that answer your question? All right. So this week, let's just stop right here. This week we're going to do some stuff on the spinal cord tomorrow and Friday. And we'll start, and there'll be more LT assignments that are due. And those LT assignments for Unit 4 will be due before Thanksgiving. And then after Unit 4, right, there's no more grace period for late assignments. There's no more, right? So you just get zeros and or whatever you get, okay? So do you guys have any questions for me? Just left.
We were born just to suffer, and we suffer just to live.
Thank you. Thank you, Tony. And
You're
thank you,
welcome. I guess. No, I'm just kidding.
And may God bless America.
Oh.